Namaste students, myself Jvwu Kumar Yadav, Faculty of Education and Methodology, Jvwu Jaipur. I have prepared this lecture for BSc B.Ed ZBC 8th sem students. Uh, Subject name is Botany, Biochemistry. Students, this is the last topic of this section. Since last couple of days we have studied protein synthesis, all those steps, their proteins structures, and all those conditions, how these proteins are stabilized, synthesized, transcription, translation, all those topics we have covered so far. This is the last topic of this chapter. This is the some of the protein inhibitors which inhibit the synthesis of proteins in living organisms. So what are those? I have selected only few of them. There are many more. Okay, I'm not going to into the detail of those, but at least being a biochemistry student, you must know few of them. So let's start. This is the schematic diagram of ribosomes, which is comprised of both the units 50S and 30S. This is the prokaryotic. Okay, so some of the inhibitors like chloramphenicol is there, which prevents peptide bond from being formed. Okay, this prevent the synthesis of peptide bond. Next one is tetracycline, which inhibit the binding of a small unit of ribosome, that is 30S. Actually, this block the attachment of transfer RNA to the ribosomes. This prevents to the synthesis of peptide bonds. Another is streptogrammains, which interfere with the distinct step of protein synthesis. Actually, this inhibit the joining of large subunit. Okay, in your competitive exams, most of the time they used to ask which inhibitors going to bind which component. So at least you must remember which portion is going to be blocked by which inhibitors. So some of them you must have to mug up anyhow for your competitive exams. Next is some of the others inhibitors are like that chloramphenicol, which binds to 50S portion and inhibit formation of peptide bond. Next one is erythromycin, which binds to 50S portion and prevents translocation, that is movement of ribosome along the messenger RNA. Another one is tetracycline, that interfere with the attachment of transfer RNA to messenger RNA ribosome complex. Streptomycin is another inhibitor that changes the shape of 30S portion, that causes code of messenger RNA to be read incorrectly. So these are some of the inhibitors which are very important for your examination point of view and even your competitive exams. Most of the time they used to ask which inhibitor is going to block which step of protein synthesis. So being a botany student, being a biochemistry student, you must know. So students, in this way we have covered our protein synthesis, protein portions. In next periods, in next lectures we'll study something about a new topic, that is fat, lipid. So we'll see all those facts about that. Okay, thank you very much.